Welcome to your prep screencast on Chapter 9. Let's get started. Okay, let's open up this here. So I'm going to give you as quick as a little review over our muscle tissue here as I can. So we're going to start off in distinguishing the differences between our three types of muscle tissue. We have our skeletal tissue that's going to be multinucleated, striated, and it's going to have voluntary muscle control. We have our cardiac muscle tissue. Of course, you can find that in the heart, and we're going to have striations here too, although you can see it's a little bit lighter than skeletal muscle. They can have 1 to 2 nuclei, and we're going to have this muscle be under involuntary control. Our smooth muscle has only one nucleus, and it's centrally located within the muscle cell. No striations here, hence the name smooth muscle, and this is also involuntary, and we're going to find this in hollow organs. I just want to mention some terminology. Myo, mice, and sarco are always going to refer to muscle. For example, we have sarcoplasm, which is going to serve as our muscle cell cytoplasm. More on that a little bit later. All right, let's move into, this is just a little chart showing us all the differences amongst our muscle, basically what I mentioned in the previous slide. Okay, now we need to talk about how muscle is organized. So first we have surrounding the entire muscle itself is going to be the epimysium. This is made up of dense regular connective tissue, and like I said, it surrounds the entire muscle itself. Now, we can go into smaller bundles, which we'll learn about in a moment. These are called fascicles, and you see them right around here. So we have a bunch of fascicles that are going to make up the entire muscle. And surrounding this fascicle is a perimysium. So the perimysium is a fibrous connective tissue. And like I said, it groups these muscle fascicles. Now let's break down the muscle fascicles, okay? The muscle fascicle has within it a bunch of muscle fibers. So each one of these circles is a muscle fiber, and each one of these muscle fibers is surrounded by an endomysium. And the endomysium is made up of a fine areolar connective tissue. Now, within each one of these muscle fibers, we're going to soon learn about these myofilaments that are going to go within each single, or myofibril is another name for it, which is going to kind of make up each one of these cylindrical portions here, okay? So let's continue on. And this is just a little chart summing up what we just talked about, so let's review. Surrounding the entire muscle itself is epimysium. Within the entire muscle, we have a bunch of fascicles. Surrounding the fascicle is going to be a perimysium. So we see a close-up of a fascicle here. There's that perimysium surrounding it. And then inside the fascicle, we have a bunch of muscle fibers. Within the muscle fiber, or surrounding the muscle fiber, let's talk about that first, is going to be the endomysium. So here's the endomysium. And this muscle fiber, by the way, is what we're talking about when we say muscle cell. So you can see here is our muscle cell, and in skeletal muscle, this is multinucleated. So we have a nucleus here, a nucleus here, and a nucleus here. And all of this is surrounded by the endomysium once again. Now we could see something here called a sarcolemma. Sarcolemma, sarco meaning muscle, and lemma referring to the plasma membrane. So this is the plasma membrane of the cell. Okay, within the muscle fiber, we have myofibrils, and we're going to take a closer look at what a myofibril is in just a moment. But first, let's talk about muscle attachments. In lab, we already talked about what a origin and insertion is, and there's a different way of discussing these attachments as well, and that's direct and indirect. So direct, or a fleshy attachment, is going to be when that epimysium, remember that surrounds the entire muscle itself, is fused to the periosteum of the bone, the outer layer of the bone, or the perichondrium of cartilage. Perichondrium, that just means the connective tissue that surrounds the cartilage. Okay. Indirect is when we have a connective tissue wrapping that extends beyond the muscle as a rope-like tendon. So we see that down in here, okay, that rope-like tendon there. Or a sheet-like aponeurosis. Remember in class I reminded you that aponeurosis means a flattened tendon. So we have an aponeurosis on the bottom of our foot here, and that is later attaching to bone. All right, so let's talk a little bit about microanatomy when it comes to our muscle fiber. Remember that also means a muscle cell. So first thing I want to discuss is that sarcolemma. I mentioned it in the 2 slides before. Remember, sarco means muscle and this means plasma membrane. So this is the plasma membrane of the muscle cell. So that really is kind of surrounding it. Above the sarcolemma would be that epimysium. Okay, they're 2 separate different things. Next, let's talk about the sarcoplasm. Now, the sarcoplasm means the muscle cytoplasm. So that kind of surrounds all of these myofibrils. Okay. Next, we have our sarcoplasmic reticulum. Okay, let's pick back up. Sorry about that. Our sarcoplasmic reticulum is where we're going to store our calcium. So I want you to look and see that each one of these is going to be a myofibril. And so they each have this network of, it's really within a cell, the smooth ER. Okay, so remember back to our cells, how it's made up of a bunch of organelles. The smooth ER, the smooth endoplasmic reticulum, is called the sarcoplasmic reticulum in a muscle cell. It's the same exact thing. And so it has all these tubules here. There's my mouse. All these tubules here in which calcium is stored until it's ready to be used. They run longitudinally along these myofibrils, and they form something known as a terminal cisternae, which, do I have a label for that? I don't think so. Okay, so hang on one second. We'll talk about T-tubules in a moment. So really this ER is going to regulate when the calcium is released and when it's stored, okay? All right, now we could talk about our T-tubules. So our T-tubules, you'll see they're kind of in between the SRs, the sarcoplasmic reticulum. So here's another T-tubule here, and it dips down, okay? So this would be the SR, the sarcoplasmic reticular here in this image, this light blue. And so our T-tubules, they are protruding down from this sarcolemma, and that's kind of how they're forming a T. And they are going to go down to our myofibril to a specific area called the A and I band junction. We're going to talk about that in the next slide. But really what we want to know here is that this T-tubule and the SR on both sides here, okay, this portion is known as the terminal cisternae. They're going to form something called a triad. And the triad is an area that's going to kind of work together in order to release the calcium into the T-tubules and for it to go to our myofibril. So we'll talk in more specifics in a moment. But before we do that, I want to talk about our myofibrils, okay? So our myofibrils, they're made up of a bunch of different parts, and we have our dark A band, and that's a little overlapping there. So it's a darker portion there, and the lighter I band portion, okay? So our A band, and by the way, I'm trying to figure out where we got these names from. So I haven't figured it out yet, but I will find out for you. But you'll see that within this myofibril, all of these different parts of it has a letter that's going to distinguish a different part of it. So one myofibril is being pulled out here is made up of several different sarcomeres. So here is a sarcomere, and it goes from Z disc to Z disc, okay? Within this, we have I and A bands. So I just wanted to point that out before we talk about A and I bands. So these A and I bands, they kind of create that striation look And an A band is representing our thick filaments, which I'm going to give you a better look at what this thick filament is in a moment. But look at this red thick line here from here to here. That's all of our A band. So that means all of that thick filament is in this dark A band here, 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 and here. Now the lighter portions is made up of the thin filaments, although our thin filaments do overlap over our A band. So we're going to call that area something different in just a moment. I'll show you. So A band, where the thick filaments are, I band, where only the thin filaments are. That's your take-home message right now. And like I said, a sarcomere, that's the functional unit of muscle, will go from Z disc to Z disc. Okay, so one myofibril is made up of a bunch of sarcomeres. Okay, so let's talk about our thick filaments. Our thick filaments are actually made up of myosin, well, it's a protein, but they're called myosin myofilaments, and they extend through that entire A band, okay? And they're going to have these little heads that stick out. These are our myosin heads. We got that blob, okay, all over here. These are all myosin heads. Those are going to act with, oh, there's our A band, with our thin filaments, which are made up of another protein called an actin filament. Or the actin filament has a protein within it, but this is a myofilament here, represented by the blue lines here as well. And then we have our Z discs. No, that's not what I'm representing for you. I'm representing in this purple box the I bands, where we have only our thin filaments showing, and it stops at the thick filament, which would make up our A band. There's our Z discs. Our Z discs are shown in green, and that is kind of holding on to our thin filaments here, okay? Our thin filaments will move in toward one another when a muscle contracts. So we'll take a look at that in a moment too. We have our M line, and our M line is representing for you, well, actually, now that I look at it, this is representing your H zone. Sorry, I don't have the zone labeled for you, but it's up here if you can see it. And this is when our thin filaments end and our thick filaments are overlapping, okay? And this H zone disappears when muscle contracts. So we have this lighter region here in the middle of our A band. Okay, so we got our thick myofilaments here and no thin filaments. Now our M line, there we go. Now we can really see our M line really well. Our M line is a line of a protein known as myomycium. I'm guessing that's why they called it M line, because of the myomycium, starts with an M, and this bisects our H zone vertically. So it just splits that H zone in half. Okay, next we got more pictures. There's our M line again. I wanted to show you, here's our thick filament made up of our myosin heads, and these are all going to attach to our actin filaments or actin molecules here in just a moment when we're trying to contract. I put this picture here because I wanted to show you a cross-section of our sarcomere. So we can see the arrangement of our thick filaments surrounded by thin filaments. Each one of these is a little bit different. So our I bands, we know that that is representing only our thin filaments. So that's the only filament we see here. Whereas our H zone, we know our thin filaments aren't overlapping there. Here was our H zone in this area. So we only see our thick filaments there. Now our M line, we see our thick filaments linked by accessory proteins, and that's what these vertical lines are representing for us. And then we have the outer edge of our A band, which would be right in this area, where we would see an overlap of our thick filaments and our thin filaments. So you see that down in here. Okay, just a quick little look at our myosin heads. These are actually made up of a bunch of tails and heads. So here is our tail of our myosin, and these would be our myosin heads, and they have these binding sites that are going to bind to actin, and I'll show you what that looks like in a moment. Let me show you this first. So here is our actin, or our thin filament molecule. These blueberry looking like substances, that is our actin subunits, and they each have active sites for the myosin heads to attach to them. Now we have also tropomyosin that's blocking all of these active sites. Do you see how it's covering those holes? Well, they're not really holes, but depressions, I should say, for those myosin heads. And then we have tropomyosin here. Now, when we have calcium binding to tropomyosin, it's going to help shift the tropomyosin strands so it reveals these active sites, and the myosin can then bind to the active sites. Okay? And these are also called G-actin molecules, these blueberry looking like structures here. Okay, so let's get back to our SR and T tubules. Okay, just to refresh your memory, here's the cisternae. We have our tubules sitting in between here vertically, and then I have a couple pictures representing this so you can see it in multiple different ways. So what happens is we get a neuromuscular junction here. We get the acetylcholine neurotransmitters going over, activating a nerve impulse, telling the muscle to contract. That information is going to travel here along the T tubule, tell the SR to release calcium, and that calcium binds to, remember troponin on our thin filaments, it binds to troponin. That's going to shift this tropomyosin strand out of the way, allowing the active sites to be attached to the myosin cross bridge. And this is just another view of those structures here. Now this is going to go a little specific. I wanted to get into this more in this prep screencast, but I want to make this short and sweet, so we're going to skip over this. We'll talk about this in class. Okay, what I want to talk about next is the sliding filament model. So I kind of mentioned this a little bit before, but what's going to happen within our sarcomere, okay? We are going to get a nerve impulse telling our muscle to contract. So when that happens, first our myosin cross bridge that has already attached here is going to release its ADP and phosphate, and that causes this myosin head to do a working stroke. So it's going to bend the head and pull on the actin filament, that's the blueberry-like structure, and slide it toward the M line. So the M line would be right here. Next, what happens is a new ATP is going to come and attach to the myosin head in order to detach the myosin head from the actin filament. After that happens, we get ATP hydrolysis, breaking off one of those phosphates from our, what was before the ATP, and now we have ADP. And so we get the myosin head cocking back and gets ready to attach once again. So when this sliding filament happens, it brings these actin filaments more in. Do you see how they're further apart here? Our H zone is bigger here. When our muscle contracts, that H zone disappears, like I mentioned before. So those actin filaments are being pulled in. So the myosin heads kind of pull onto it, in, pull in, like that. Okay, so nerve stimulus junction. Now, what happens here is we get an action potential, or a nerve impulse sent down through our nerve. We're going to talk more about that in our nervous system. And then we're going to get vesicles here toward the end filled with acetylcholine neurotransmitters. These neurotransmitters are going to get released when we get calcium entering into our axon. That again is causing the vesicles to come up at the end here, releasing the acetylcholine through exocytosis, and then they'll attach to our receptors here along our neuromuscular junction. This is also called a motor end plate on our muscle. Okay, so at this site, we zoomed in here for you to see, we are going to have our acetylcholine causing sodium to move in and potassium to move out. When that happens, we get a action potential. Our acetylcholine is then going to be terminated by either degrading the neurotransmitter through an enzyme called acetylcholinesterase, or or the ions just start, or the neurotransmitter rather, is going to start floating away from this area, or it gets used up here at our acetylcholine receptors. Okay. So like I said, this is our end plate, motor end plate here. And so we're going to go through a couple different steps. We have our end plate potential, which basically is our action potential taking place. So we get our acetylcholine that is released from this neuron here, and then it's going to cause these chemically gated ion channels to open up. When that happens, and I think I have this in the next picture, yes. So when that happens, we're going to get our sodium moving through into our muscle, and potassium is going to float on out. So here's our next step. This floating of the sodium moving on in, that is depolarization. Before I go into this in more detail, let's talk about a potential in here. Usually a muscle cell is going to be sitting at -60 or -70 millivolts. Okay, so that is the potential inside the cell. Now when sodium moves in, that causes the inside of the cell to become more positive. So that means we're moving away from -70, let's say, and moving closer to -50, -40, -30, 0. +15, +30. Okay, so we're moving more positive. When that happens, it causes an action potential to take place once we get to a specific threshold. We'll talk more about a threshold in a moment too, but once it gets to a specific level, we're just going to say +30. That's when the action potential or the impulse gets sent through, and we're going to get the muscle contracting, and then we're going to get an opening of our potassium channels, and potassium is going to float out, and that causes the inside of the cell to become negative again, restoring what that potential was to begin with. Okay, so we get more of a negative state on the inside of the cell. This is all of that summed up in a chart, excuse me. So in this example, we're at -90. Okay, this is inside the muscle cell. Our sodium channels open up. That's causing the inside of the cell to become more and more positive, which is called depolarization. And then we hit our threshold. When we hit our threshold here at +30, that's going to cause a muscle contraction. When that muscle contraction occurs, it's done its job, and the sodium channels can close, potassium channels open on up, and we start becoming more and more negative inside of the cell. So potassium's exiting, and we get back to our negative state, and the potassium channels are then going to close. Okay, so this is kind of putting the whole picture together for you. I'm going to talk about this more in class, so we're just going to skip this right here. Again, we're going to get our impulse being sent through, okay? And then we get our calcium that's released from our sarcoplasmic reticulum moving into the T tubules. When that happens, we get all of these calcium cells. Sorry, I think I said calcium into the T tubules. The calcium moves into our sarcoplasm, which will bring this calcium to our myofibrils and can attach to our troponin, which causes the tropomyosin to shift out of place, revealing the active sites of our actin, G-actin molecules here. And then the myosin heads attach and cock their heads and cause a stroke to happen. And that is when our muscle is going to contract. Okay, this is just a little review of all that. I really liked this chart. I pulled this out of a physiology text that I got. So this will take you step by step through all of the process of basically how the action potential causes the calcium to be released and where it binds and everything. Okay, motor units. This is a motor neuron that is going to basically each of our muscle fibers. So each muscle fiber has its own motor neuron. So based on how much we need a muscle to contract, it can cause a muscle to contract here and there. Okay, I think I'm going to stop before we get into muscle twitches and everything. I just wanted to give you a quick little look into Chapter 9. We're going to talk more about this stuff in class. It's basically tied into getting the inside of that cell more positive to reach our threshold, and then once we get that threshold, that's when we're going to get these muscle fibers contracting. And the more and more stimuli that we get, then more and more fibers are going to be recruited to contract. But we of course have a maximal stimulus. So that means we can only contract a muscle so much. And after that, all of the fibers are being used. So we can't go beyond that, even though the stimulus might be stronger and stronger. Okay, and these are just different ways in muscle contracting. So like I said, we'll talk about this in class. We also are going to have muscle responding to change depending in the stimulus strength. So when we get a small stimulus, we will get smaller muscles or smaller fibers contracting first. And then as we get more of a stimulus, we need to contract more muscles. We're going to pull in our larger and larger muscle fibers. Now there's different types of contractions. We have concentric contraction and eccentric contraction. These both fall under the topic of isotonic contractions. So what a concentric contraction is, it's basically what you truly think of when you think of contraction. The muscle is shortening and doing work. So think of like your biceps brachii flexing the elbow. That muscle is shortening, causing that action. Whereas eccentric contraction, that's going to kind of do the opposite. That is going to cause the muscle to lengthen. So you want to think of your biceps doing a concentric contraction, causing a flexion at the elbow, but our triceps brachii, that's doing an eccentric contraction. That one is lengthening while our biceps is shortening. Okay, and within these contractions, the load is going to be greater than the maximum tension muscle can generate. So muscle is neither going to shorten or strengthen when it comes to isometric contractions. Oh, sorry, getting my slides mixed up here. Okay, also muscle metabolism, we'll talk about that in class because that's going to take some time. And we'll talk more about these different types of muscle fibers in class as well. I think this table does a really great job summing up the differences between the three different types of muscle fibers. We have slow oxidative, fast oxidative, and fast glycotic fibers. Basically, each of these different types are going to vary as far as whether oxygen is present or not. So anytime you see oxidative, that means that we have oxygen present. Anytime you see glycolytic, that means oxygen isn't present and we need to go through our anaerobic pathways, and those pathways we'll discuss in class. Okay, smooth muscle. Like I said, we find this in the walls of our hollow organs like blood vessels, our stomach, intestines, and our heart is the only one that's going to contain cardiac muscle, even though it's a hollow organ that's made up of our cardiac muscle. So these muscle fibers are a little bit different. They are spindle-shaped fibers, meaning they're kind of fat in the middle and they taper out on the outside. They also have that nucleus centered in the center. So we find that right in the middle. So that's really going to distinguish for us that this is smooth muscle, as well as having no striations. We only have an epimysium in our smooth muscle as well. So they have very different arrangement. Also within our smallest blood vessels and hollow organs, we're going to have two sheets of smooth muscle. We have a longitudinal layer and a circular layer, and I'll demonstrate this in class for you or give you some examples. But our longitudinal layers, that's going to look like this in cross-section of a histology plate, and that's because these fibers are coming toward you, okay? These fibers are coming toward you, whereas our circular layer, these are kind of moving around in a circle like this, and that's why we see them paralleled up like this, but this would circulate around the way we see it in this intestine here. So the difference between this is when these longitudinal muscles contract, that's going to cause this tube, the intestines, to shorten. So take a tube, think of it as the intestines, and then shorten that up. So that's our longitudinal layer at work. Whereas our concentric muscles here, similar to like our orbicularis oris or orbicularis oculi. When this muscle contracts, it's going to close up this lumen. So think of your mouth. When you are making that kissy face, you're closing off your mouth, and that's what it does here in the intestines when this circular layer contracts, and this is called peristalsis. So it alternates contractions, these circular contractions throughout the intestines in order to move the food throughout. Okay, also the nerve supply is going to be different in our smooth muscle as well. We're going to talk more about this in class, but we have things called varicosities, and these release the neurotransmitter in a wide synaptic cleft here. This is the microscopic look to smooth muscle. We'll talk about this in class too. And instead of having tropomyosin and troponin, we have something called calmodulin. It's going to contract a little bit differently too. We have a different setup of our actin and myosin, and that is our quick prep of Chapter 9. Again, I'm trying to make them as close to 15 to 20 minutes. We went over again, but we'll discuss this further in detail in class.